Hello everyone, welcome to 10-Minute Physiology. In today's video, I'm going to give you a short explanation as to how skeletal muscles contract. So with that, let's begin. So I'd like to start off by first describing to you the different proteins that are involved in the sarcomere. So the sarcomere is the contractile unit of muscle. So it is going to be responsible for contracting muscle. So the sarcomere is what's going to govern muscles contracting, and therefore it governs how our different body parts are going to move. So it allows our arms to do a bicep curl or us to do squats or whatever. So the sarcomere is going to be the contractile unit of the muscle. So the sarcomere is actually going to be highly organized, as we see right here, and it's going to consist of many different proteins that are organized in a specific way. So the first component of the sarcomere I really want to talk about is something called the Z-line. So the Z-line is represented by this line of yellow circles here. And the Z-line or Z-disc is going to be made up of alpha-actinin. So alpha-actinin is the protein that makes up the Z-line. Now the Z-lines are important because they are what determine how long a sarcomere is. So the distance between one Z-line and another Z-line is going to determine how long a sarcomere is. So as we see right here, between this Z-line and this Z-line, we have one sarcomere. Now the next important protein that is in the sarcomere is going to be actin. So actin in its monomeric form is going to be globular, but actin can come together to form fibers. And these fibers are helical fibers, as we see right here. So these actin molecules, which make up these actin fibers, are going to be making up the thin filaments. So the thin filaments are going to be largely comprised of actin, and along with actin, we have a bunch of other proteins. So another protein that is part of the thin filament is going to be nebulin. So nebulin is sort of like a molecular ruler. It sort of acts as a spring that prevents sarcomeres from expanding or contracting too far. So now we're going to look at the thick filament, and the main component of the thick filament is going to be myosin. So myosin, as we're going to see, is going to be the main driver of contraction. Now, along with myosin in the thick filament, we have titin. So titin is another protein that is very similar to nebulin in that it acts as a molecular ruler. So those are going to be the main proteins that you have to know in order to understand how the muscle contracts. So now let's talk about the different bands. So there are several different bands that are in the sarcomere. And the first band is actually going to be the A band. So the A band is going to span the entire length of the thick filaments. So the A band measures the length of the thick filaments. So the next band is going to be the H band. So the H band is going to only consist of thick filament, as we see right here. The next band is going to be the M band, and the M band is going to basically contain proteins that connect the thick filaments together. And in the middle of the M band, we have the M line, which is the middle of the sarcomere. Now, along with that, we also have the I band. So the I band only consists of thin filaments. So these are going to be the major bands, and these bands are going to become incredibly important when we're talking about how the sarcomere looks when it contracts. So we're going to come back to this a little later in the video. So now that we know the different proteins and the different bands that are in the sarcomere, let's talk about the different filaments, and we're going to start off with the thin filaments. So this right here is a picture of the thin filaments, and what we're going to do first is identify all the proteins that are involved in the thin filament. So the first is, of course, going to be actin in its fibrous form, so actin that comes together to form a fiber. You also have, though, some other important proteins in the thin filament. So one of these proteins is going to be tropomyosin. So tropomyosin is going to be incredibly important for regulating when contraction occurs. And we're going to talk about that in a second. So along with this protein, we also have troponin C. So troponin C is going to be responsible for binding to calcium and therefore initiating contraction. And along with troponin C, we also have troponin I, which basically acts to inhibit contraction. And we also have troponin T, which basically connects to tropomyosin and it basically moves tropomyosin around. So how do all these things come together? So let's just say that we have a bunch of calcium flow into the cell. So we have calcium that flows into the cell. So as we talked about in previous videos, calcium is going to initiate contraction. So calcium, as we said, is going to bind to troponin C. So calcium comes in and binds to troponin C, and this is going to initiate a conformational change in the troponin complex. So the troponin complex is going to be the complex of the three troponin proteins that we talked about: troponin C, troponin I, and troponin T. So when calcium comes in and binds to troponin C, it'll cause a conformational change, which basically initiates the contraction cycle. So what's going to happen is that troponin I is going to be moved out of the way by troponin T. And troponin T is also going to move out of the way tropomyosin. So tropomyosin, which we talked about, is a protein that covers up a specific region on actin molecules called the myosin binding site. So the myosin binding site is basically where actin and myosin come and bind together. And when they bind together, this allows the cross-bridge cycle to occur, which therefore allows contraction. So when calcium comes in, it binds to troponin C, which basically initiates troponin T to move troponin I out of the way, and troponin T also moves tropomyosin out of the way, which therefore allows the binding site to be opened and allows myosin to come in and bind to the binding site. So when calcium decreases in the cell, calcium will basically unbind or dissociate from troponin C, which will basically cause troponin I and troponin T to move back to their original places, and then it will move tropomyosin back over the binding site, which will therefore prevent cross bridges from being opened. So that is going to be the thin filament. So now that we know how the thin filaments work, let's talk about the thick filament. So just in case, though, you didn't get what I said, here is a little worded explanation of what I just said. So now let's talk about the thick filaments. So the thick filaments look something like this, and they're made up of myosin. So the structure of myosin is this. So the first part of myosin is the head, and the head of myosin is basically where all the action is going to be occurring. So this is what we're going to focus on in this video. So the head of myosin is going to have ATPase activity. So it's going to hydrolyze ATP and use that in order to contract muscles. Now, along with the head of the heavy chain of myosin, or myosin heavy chain, we also have the tail regions of the heavy chains. We also have the hinge region, and we also have two light chains. Now, we're not really going to be focusing on these other things. We're mainly only going to be focusing on the head. And that's because the head is going to be where the ATPase activity is, and it's going to be where interaction between actin and myosin is going to be occurring. So now let's put these two filaments together and basically see how they interact in order to produce contraction. So the process of contraction occurs with something called the cross-bridge cycle. So the first step of the cross-bridge cycle is what's known as the Riger state. So the Riger state basically is when you have a myosin head attached to the actin molecule inside the thin filament. So in the Riger state, the myosin head has a very high affinity for actin. So in order to start contraction, you have to decrease the affinity of the myosin head for the actin molecule. So in order to decrease the affinity, you have to bring in ATP. So ATP is going to come in and bind to the myosin head and decrease the affinity of it for actin. So basically, it's loosening the cross bridge. So what happens after that is that the myosin head, as we said, is going to hydrolyze the ATP because it is an ATPase. So when it hydrolyzes ATP, it's going to produce ADP and inorganic phosphate. Now, both of these things are going to remain bound to the head. And after this hydrolysis occurs, it provides the myosin head with enough energy in order to go into what's called the cocked state. So what we see here is we started off in this position, and then after the hydrolysis, we get this position, which is referred to as the cocked state. So after this occurs, what happens is weak cross bridges form. So basically what we see here is that the actin head moved from this actin molecule, which was in blue, to another actin molecule, which is in yellow. So it moved from one to another. Now, since the phosphate is attached to the head, the cross bridges that are formed between the myosin head and the actin molecule are going to be weak. So in order to strengthen them, you have to remove phosphate. So phosphate comes off of the myosin head, which allows strong cross bridges to be formed. So once the phosphate is removed, it allows the myosin head to undergo a conformational change called the power stroke, which we see here. So you see it goes from this cocked state to this state. And by doing this power stroke, what it does is it actually slides the thin filament this way. So it slides the thin filament this way, therefore shortening the sarcomere. So this power stroke is what causes the thin filament to slide against the thick filament and therefore produce contraction. So after this occurs, what happens is that we have to start the cycle over again. So what happens is that ADP will leave the myosin head and then it goes back into the Riger state, which allows the cycle to occur over and over again, producing contractions. So that is the cross bridge cycle. So now that we know how contraction occurs, let's talk about how the contraction affects the sarcomere itself. So this right here is a visual representation, a very simplified visual representation of two sarcomeres. So just for repetition, let's just label each of these parts. So these lines are the Z lines. This right here is the thick filament. So therefore the length of that filament is going to correspond to the A band. This area is called the H band. And remember, this is going to be a region in which you only have thick filament. This region is the I band, and this is the region in which you only have thin filament. And the region between two Z lines is going to be one sarcomere. So this is just a simplified representation of the sarcomere in order to understand a specific concept. So basically what we're going to show is what happens when this contraction occurs. So what happens when the cross bridge cycle occurs? So remember, in the cross bridge cycle, when we get to that power stroke section, what we see is that the thin filaments slide against the thick filament. So how does that look in a sarcomere? So let's say we start off in this relaxed state. And let's say that after a contraction, it looks something like this. So what sort of changes do you see in the sarcomere? So the first change that you should see is that the length of the H band has decreased. So remember that the H-band is a region in which you only have thick filament. So what we see here is that since the thin filament slid against the thick filament during contraction, the amount of area or the length in which you only have thick filament has decreased. So the H-band has decreased during the contraction. Also, the I-band has decreased during the contraction. Another thing that decreased during the contraction is the distance between two Z-disks or Z-lines. So during a contraction, the sarcomere shortens, which therefore means the Z-lines are brought closer together. Now, the only thing that doesn't change length during a contraction is the A-band. And the reason why is because the A-band measures the length of the thick filaments. Now, during the contraction, what happens is like we said, the thick and thin filaments slide past one another. They don't contract in length themselves. They just slide past one another. Therefore, the length of a particular filament is going to remain constant. So therefore, since the thick filament remains the same size over the course of the contraction, the A-band is also going to remain the same size. So remember that during a contraction, all bands except the A-band decrease in length. And even the sarcomeres decrease in length. And this shortening is what's going to produce the contraction that we see in our muscles when we move them. So in summary, we talked about the sarcomere structure and that it's the contractile unit of the muscle. We talked in great detail about what the two filaments were that make up the sarcomere. We talked about the cross-bridge cycle, and we also talked about how the two filaments slide past one another during a contraction. So I hope this video helped you understand how skeletal muscle contracts, and I hope to see you in the next one. Thanks for watching, and I hope to see you next time.